We have our next speaker, Emma Teeling, who will, I wonder what she's going to talk about. I don't know, actually. Let's see.
First of all, I want to say thank you so much to the conference organizers. It has just been a privilege to be here. This has been unbelievably exciting. And to hear all of these new advancements in the field of aging research was extraordinary. But I want to take a slightly different approach to this. And I want to talk about the most fascinating, the most brilliant, the group of mammals that can save all of us. And these are the bats. So does anybody know what species this is? The fingers might be a giveaway. This is the smallest mammal in the entire world. This is Crassionyctus tonglongii, or the bumblebee bat. They're the fingers of my very first PhD student holding it. So bats are extraordinary. They're very, very small. But studying bats can allow us to address two of the greatest challenges that society faces today. And the first we know is really how we can age more healthily. It's been the whole point of this conversation. But another part is how can we deal with infectious disease? And if we want to have healthier lifespans, we're going to have to find ways that we can deal with disease. Because aging is the damage of living, but also you're exposed to pathogens. So you have to find ways to deal with infectious diseases. And unfortunately, this has really been brought to all of our consciousness with SARS-CoV-2. If you think about it, there's recorded 6 million deaths, estimated probably 20 million deaths in 24 months from a new zoonotic virus. But just imagine if that virus had evolved slightly differently. If you had the infectivity of SARS-CoV-2 with the lethality of MERS, another zoonotic coronaviruses, we wouldn't be back on our feet yet. So we need to find ways to deal with infectious disease and also to find ways that we can age more healthily if we want to have longer health spans. But how are we going to do this? So I'm a zoologist, first and foremost, and I love this book and I highly recommend that everybody reads it. And it's a brilliant book by Steve Ousted. And really, Steve Ousted's research stimulated me to think about bats in a slightly different way. But I want to read an excerpt from it. Recall that nature, in the guise of certain animals such as birds, bats, and mole rats, has repeatedly discovered how to deal with damaging free radicals much better than humans can. Other species, like elephants and whales, have developed dramatically better cancer resistance than humans, and so forth. At some point, I am confident the full armamentarium of the biomedical research enterprise will be deployed to study and eventually understand these lessons nature has to teach us about preserving and prolonging health. And so with all of these new technologies, we're now allowed to think of and imagine studying these processes in non-model organisms that potentially are better model organisms to study extended health span. And I'm going to talk about these beauties. So 1 in 5 of every living mammal on this planet is a bat. There's lots and lots of them. They're found throughout the entire globe. And the one thing that bats do that no other mammal does is that they fly. All bats fly. Now, flight is very, very metabolically costly. Think how difficult it is to evolve as a form of locomotion. But really, bats expend up to 130-fold times more oxygen, they consume it by flying. And flight is very metabolically costly. They're the only true flying mammal. This means that bats have had to evolve ways to deal with this deleterious effect of a really, really high metabolic rate and also small body size. But it also allows them to get all around the world. Also, they use laryngeal echolocation to orient in their environment. And if you think about it, bats can live for an extraordinarily long time, yet they can't go deaf because they won't be able to move around. So studying bats will allow us to understand how we can hear better into older age. But they have a unique immunity, and they're able to tolerate the likes of serious pathogenic diseases, such as Ebola, Marburg, SARS and MERS-like coronaviruses, potentially rabies, without getting sick. And the question is, what's the relationship to COVID-19? And I'm going to talk a little bit about it, and how can they save us? But for this conference, the thing that really intrigued me about bats was that although bats are some of the smallest mammals in the entire world, they are the longest lived mammals given their body size. And this bat holds a record. It's Myotis brantii. So this bat was caught as an adult, a member of this species, was tagged, was released, and was caught 41 years later with little to no signs of aging. And the 41 isn't the big deal. The big deal is that this bat weighs 1/3 of a lab mouse. So how do they do this? Because typically in nature, and again, this comes from Steve Ousted's work, you can predict how long something will live for given its body size. Big things live slow and live long. Think of bowhead whales. Small things live fast and have short lifespans. But bats buck this trend. Indeed, according to this paper, there's only 19 species of mammal that live longer than a human. 18 of these are bats, and one's a naked mole rat. And Vera's going to be talking about that. So bats seem to have evolved mechanisms to allow them somehow resist and find ways to deal with aging. And so what I want to do is I want to talk to you about the work I've been doing now for the past 12 years, about trying to develop bats as a unique model system to understand how we can age more healthily and have better immune tolerance. But how do you do that? Well, the first thing you're going to need if you want to work in a non-model organism is you need a good genome. And when I started this all out, all the bat genomes out there were just little pieces. And it was very, very difficult to work with this. So what do? You got to go and you got to make them yourself. So myself and Sonia Vernes are both the founding directors of this big genome consortium called Bat1K. The idea is that we want to sequence the genome of every living bat species to chromosome-level reference quality genomes. We have some amazing scientists, computational biologists, technologists, all involved in doing this. And we were able to publish our first pilot study, which were 6 reference quality bat genomes in Nature in 2020, showing that actually we can find a way to be able to sequence these genomes, work throughout the entire world collecting bats, and we can analyze them to pull out the beautiful juice and stories in their genomes. So far, we have our pilot study is done. We have a representation now of every single bat family, which is difficult. Some of these bats are only found in these tiny regions in Thailand, for example. We're looking to move to phase 2 and phase 3, whereby we really want to sequence these bat genomes to try and understand what are the adaptations that bats have evolved, what are the molecular bases of the unique adaptations. So you need genomes. But how do you study bats in an aging context? So think about what you do, what everyone's talking about with their model organisms. You can study a mouse in the lab throughout its entire life. Take samples whenever you want. But the problem with trying to study these long-lived bats is that they don't do well in captivity. They have 1 baby a year. So how are you going to study bats in an aging context? Also, they're small. How can you take these non-lethal samples from bats? So I'm going to show you videos I talk through this. So this is a population of bats that I've been studying now for the past 10 years in Brittany and France. So we're asking the question, we need to find a population of the longest-lived bats, and these are the Myotis genera, that are large enough that we can take non-lethal samples, which means taking up to 140 microliters of blood, that we can take a wing punch and a tail punch, 3-millimeter punch, from the same individual year after year after year. We need a whole bunch of biological data. How much do they weigh? How healthy are they? We need to be able to catch them year after year after year. So working with Brittany Vivant, a grassroots conservation organization in Brittany, they had been studying and looking at these bats for the past 20 years. They had caught them. They had caught every single bat in 2010. They had tagged all of them, put a little microchip in. The only way that you can age a bat, if you don't use Steve Horvath's clock, is that you can open up its wing, and if you see the finger bones are not yet fused, you know that this is a baby. It's under 1. The minute that the finger bones are fused, that's it. And very, very quickly, bats go from being small babies to nearly the size of adults within about 6 weeks. They're like birds, they have to fly. So we found this population that we could catch the same bat every single year. We could take non-lethal samples. And we wanted to work out, well, what happens in bats as they age? So we want to ask the question, well, what happens with their telomeres? So we used a qPCR method to do this. Remember, we're dealing with really small samples. We want to look at their mitochondria. Do bat mitochondria show the same level of oxidative damage as would be expected given their high ROS production? Look at their blood transcriptome. As bats age, they show the same level of dysregulation as we do. Look at their microbiome. What happens to their microbiome as they age? Look at autophagy, another hallmark of aging. If there's lots of damage being because of their high metabolic rate, are they able to remove it better? And of course, look at their innate immune response. So I'm going to couple down 10 years work and 60 years of field data into a couple of slides. So we looked at how telomeres changed using this relative Cawthorne telomere method between young, middle-aged, and older bats. This was work done by Nicole Foley, led by Nicole Foley. What we found was that in the longest-lived bats, their telomeres did not shorten with age. So in all of my other bats we looked at, telomeres didn't shorten with age. It doesn't seem to be as a result of telomerase expression because we couldn't find that in the RNA from the blood. We also were able to take wing punches and grow fibroblasts in the lab. We still found no telomerase expression. So we went and we looked at our genomes. We did a whole phylogenomic analysis across multiple different mammals to see could we find any genes involved in telomere maintenance that may be evolved in bats different to other mammals. And after we did Brahms-Faroni correction, the destroyer of all data, we really found only about 2 genes. But again, these genes were involved in DNA repair. And potentially this indicated that bats may have been using this alternative telomere length mechanism. But I want you guys to think of nature, again, as experiments. Because the other thing that bats actually do is that they go into hibernation. And there's been lots of conversations about muscle atrophy. And what happens if you're put into a boot? And why after 6 weeks you need physiotherapy. But this is work done by Megan Power in my lab, looking at how telomere length changes within individuals while they're hibernating. And it's quite extraordinary because you find that when bats hibernate, it seems to be a form of rejuvenation. So this is something to look at. What about their mitochondria? According to the free radical theory of aging, there should be a huge level of oxidative damage when you look at how mitochondrial heteroplasmy changes between young, middle-aged, and older bats. So we sequenced the entire population of mitochondria in young, middle-aged, and older myotis bats. And what we found that there wasn't any evidence of this oxidative damage. So this implied that bats potentially were able to repair the damage or remove the damage. When we looked at their bat microbiome, so taking an anal swab and doing metagenomic sequencing of all the bacteria there, what we found that when you looked at young, middle-aged, and older bats, there was no change in their microbiome. They maintained homeostasis. Again, when we took this 140 microliters of whole blood, we sequenced the entire RNA that was present. We looked at young, middle-aged, and older bats, and we were looking for this age-related transcriptional changes. We also wanted to look at what was regulating this. So we looked at the microRNAs that were also found in that same sample of blood from the same individual to see could we find these underlying regulatory mechanisms, to see could we see what was happening in bats as they aged. And so what we found when we were able to compare whole blood RNA-seq between bats, healthy humans, healthy mice, healthy wolves, was a major difference was that at bats' age, they increased the maintenance of their DNA. That's what really hops out. If you look and you see mice become very inflamed as they age, and the bats are not. But what really made me feel happier with this was, people thought, that's a crazy way of trying to study aging. What we found when we looked at these samples taken in the wild, sequenced all their RNA, we found that bats naturally showed these transcription profiles that mice that you would modify in the lab do to live longer. For example, there's P10. As bats age, they increase the expression of P10 with age. And so that meant that our methods allowed us to uncover longevity mechanisms that bats may have evolved. But I remember the brilliant Luke O'Neill said to me, so what, we already know all that. What we found was potential new candidates. And these were these different genes that showed a very different expression profile in bats to the other taxa. And these are genes that we should look at and target, and they're new ones. But we also found that this was regulated by microRNAs, and this is something I think we should think about. MicroRNAs regulate a whole bunch of different genes and transcripts. What we found was that there were certain microRNAs, we've now been validating that are in silico, result was actually really real in the lab. And that potentially there are certain microRNAs that may regulate this longevity profile. And I think we should think about using microRNAs also as a druggable way of doing things. What about autophagy? When we looked at our blood transcriptomes and saw how it changed for young, middle-aged, and older bats, all of the autophagy transcripts increased with age in bats. We then went and we took wing punches and grew fibroblasts from known aged bats, so young ones, middle-aged, and older ones, compared them with mice. This work was done with Vera. What we found was that we were able to recapitulate the results. So when we starved those cells, we looked at the LC3 2-to-1 ratio, it seemed to increase with age. That was a proxy for autophagy. Again, a lab result validating a field result. We then went and looked at the genome to see could we find any genes that showed evidence of selection, and we found a whole bunch of them. So autophagy is also important in bats as they age. But again, let's think about models. So across the aging spectrum in bats, longevity spectrum, you have shorter-lived and longer-lived bats. And the shorter-lived of all bats that we know of is Molossus molossus, living for as long as expected given its body size. This is a nice experiment. What happens in shorter-lived bats? We just took some blood from these Molossus molossus in Panama, and what we found was quite interesting. When we compared the blood transcriptome of adults, what you found that there was no difference in the expression of the pro-longevity genes, but the anti-longevity genes, again, the bad, seemed to be what was different in the shorter-lived bats. And we need to look into this. What about our molecular clock? What about our epigenetics? This was work done by Steve Horvath and by Jerry Wilkinson. 26 species of bats, 700 individuals of known age bats using Steve Horvath's clock. What we found was that actually you could make a bat clock and predict the age of bats from this clock. And this is a game changer. Now we can catch a bat now how old it is. But what I found most exciting was the longest lived bats, their clock ticked more slowly, and the shortest lived bats, their clock ticked much more quickly. And the longevity sites seem to correlate with promoters involved in DNA repair, anti-tumorigenesis, and also innate immune. Something interesting, just hot off the press when we do this, what you also find is that when you look at these hibernating individuals, again with Steve Horvath's clock, you can find this kind of slowing down of aging during hibernation, and that this clock, the telomere clock and the epigenetic clock, are matching. So how do they defy the aging process? DNA repair is increased, damage is removed, telomeres are maintained, but they also have a balanced immunity. And this is something that we're going to talk about very briefly. And it's modulated by microRNAs. But very fast, we're getting on to balanced immunity. This is the one thing we found in bats, was that as they age, there seems to be this balance between these highly inflammatory transcripts and also these anti-inflammatory transcripts. And again, we talked about the fact that we're dealing with horrible times. But how are bats involved in all of this? So bats are implicated as the potential reservoirs of all the SARS-CoVs. Now, of course, there's more viruses in bats because there are more bats than, well, there's more rodents, but there's an awful lot of bats. And viruses will co-evolve to exist with their host. But the thing that's very different in bats is they have a high preponderance of coronaviruses. They seem to have evolved a unique way of living with these viruses. And so we want to ask the question, well, how do they do this and how is this related to aging? So the work that we were able to do in my lab, again, asking the question is, what is the innate immune response of bats? Caught a wild bat, this is from our study. She had to be put down. She'd lost her wing. We were able to isolate stem cells, turn them into macrophages, and do experiments in the lab with immune agonists to see what happens with bats, macrophages, as they have an infection. And what we found very quickly was bats very first made a very aggressive antiviral response, and then they matched that with an even more aggressive anti-inflammatory response that allows them to tolerate these pathogens. When you look within the genomes, you can already see this. And we asked the question very briefly and quickly, could we look in the genomes of reservoir bat species and see what they've evolved to slow down aging? And what we found is when we looked at all these known pathways in SARS-CoV-2, what we found is that there's huge selection in bats compared with other mammals. And this is something that's quite interesting. How bats respond to this, to infection. The information that we have in our genomes. A bad response to COVID-19 is like inflammaging. So potentially bats have evolved ways to deal with both things at once. And that's what we're looking at. And to leave on this kind of scary note, it is about bats. When you do look within their genomes, what you find is the highest diversity of endogenized viruses. So again, sequencing bat genomes can allow us to understand viruses they can live with. So again, flight potentially has driven all of these unique adaptations, allowing and forcing bats to evolve mechanisms to deal with this constant sterile inflammation they must experience from this high metabolic form of locomotion. I hope I've convinced you studying bats will allow us address both of these challenges. And to finish off with the brilliant Steve Ousted, Methuselah Zoo, I believe, holds the key to prolonging human health. It may seem like a radical idea, but perhaps a radical idea whose time has come. Let's all agree to acknowledge that evolution is cleverer than we are. And he said this, not me. Are you listening, Silicon Valley zillionaires? Thanks, guys.
Thank you so much, Emma. We have time for one question. Magnus, down here.
Hi. These these particular changes in bad genotypes and phenotypes, presumably that's due to lack of predation, I would assume.
Sorry?
Presumably these adaptations in the bats are due to lack of predation because they fly. This is also seen in birds, right? And that's not my question. What I want to ask is this conserved in other mammals that, like bats, experience a decrease in predation? So say whales that are extremely large, or perhaps mammals isolated on islands with no predators?
It's a good question. So you're saying, does this reduced predation allow for the maintenance of self? Is that the question?
What I'm getting at is, these adaptations, are they universal across all mammals?
No.
Okay.
So what we're seeing is that they're specific to, so we're looking for the ones that are different in the bats. Anyway, that's how we search for it. So no, they're unique to bats.
I'm sorry, but we're already past time. Emma, that was spectacular, fantastic talk. Amazing. And